Hello, it's Jim, the author behind the blog, Reverse Engineering the Origins of SARS-CoV-2. This vlog will be about a couple of Substack posts from a couple of years ago. The first one was titled SARS-2 was in a UNC freezer back in 2018, and the next one, the next week, was titled SARS-2 was patented by Ralph Baric in 2018. So there was a lot going on in 2018. Where did this outrageous claim come from? It was a relatively new FOIA request by U.S. Right to Know in late 2023, early 2024, on the United States Geological Survey for Ralph Baric's records in the DARPA Defuse proposal. So this Defuse grant proposal was led by EcoHealth Alliance, Peter Daszak, and mainly written by Ralph Baric himself because it is so technical, it's so deep, and we'll get into what it was. So as I write here, if you closely read what I tried to write, you are probably shocked at this one line from a recent 1,400-page FOIA. Ralph Baric, RB, in 2018 has already generated SARS-like chimeras of bat viruses which are 20% different from epidemic strains. What are epidemic strains? SARS-1. He basically has, in 2018, created a chimera, which is basically a combination of many viruses, think like Lego blocks, assembly of a novel new genome from a bunch of other genomes. And he has created something about 20% different from SARS-1. Well, guess how much different SARS-2 is from SARS-1, 20% different. So this is quite the shock right here. This line right here that Ralph Baric, a white man in a white lab coat on the campus of Chapel Hill, North Carolina in 2018, has created something very similar. Very similar. We're talking a few percentage points within SARS-2 in 2018. So this is why I can make the claim. SARS-2 was in a UNC freezer back in 2018. Now, it gets a little complicated in the weeds. We're trying to figure out how similar it is and what on earth it is. But if you closely read what he's got here, he says something called 293 and HK3, which he's combined together to make this chimera, are both 20% different from SARS-1, and SARS-2 is 20% different. Ralph Baric said it was 22% different. Xi Li says it's 20% different. When you get into the weeds of amino acids and nucleotides, you can get a little vague about what it is, but we're talking about something within a percentage point or two. In Ralph Baric's freezer in 2018, so there's something even closer in Ralph Baric's freezer in 2018 than Xi Li in 2018. Xi Li has RA2G13, which is about 4% different, and Ralph Baric has something within a 1 or 2% of SARS-2 in his freezer in 2018. So he's got something even closer. So Ralph Baric even testified later on when he was asked, what on earth is that in your freezer? And Ralph Baric admitted, I had something that was within the range. And what was it? It was essentially something called HKU3S mix. That's what it was. The 293 and HK3 were basically something called HKU3S mix. And you can put it together here, essentially. Deeper into the FOIA, there's 279, and then there's HKU3, and he's mixed them together to make HKU3S mix, again, a chimera. So this HKU3S mix chimera, which is in this 2018 FOIA, written by Ralph Baric, is much, much, much closer to SARS-2 than anything and anywhere else on the planet. So this is quite the switch. I could never see inside UNC's files because he's basically still, to this day, I'm recording this in 2026, he's still fighting U.S. Right to Know to not give up his files. So U.S. Right to Know, Emily Kopp, to be precise, did a FOIA move, it's called. She went to the USGS, the United States Geological Survey, who was a partner on the DARPA Defuse bid that they lost, and we'll get back to that in another post. But inside the USGS files were Ralph Baric's files, his emails, essentially. And in those emails is this shocking claim up here that he's got something 20% different. So that is quite the shock. And because you can say he's got something called HKU3S mix, which he patented in 2018, I can then follow up the next post with Ralph Baric patented SARS-2 in 2018. So this is quite the detail. I don't want to go into here in detail. You have to kind of read it yourself to get your mind around what he's up to. But there were other bombshells in this FOIA by Emily Kopp of the USGS. And in there was a woman named Danielle Anderson. This was another shocker. It was kind of vague and odd what she was doing on the DARPA Defuse bid. Because she was in Wuhan. And she was known as the last and only foreign scientist in Wuhan. And here she is in this DARPA Defuse bid. And she's going to lead the animal studies with Ralph Baric and her boss, Linfa Wang, in the Wuhan BSL-4. And she's in there in the Wuhan BSL-4 and mysteriously left in November. November 2019. She never told us what she was doing. November 2019. Her most recent stint ended in November 2019. That's an interesting time to be leaving Wuhan, and you're the only foreign scientist at the Wuhan lab? And what did she say she was working on? Not DARPA Defuse, not with Ralph Baric, not on SARS-like viruses that are 20% different from SARS-1. She says she's working on Ebola. So I'm not going to pull up her resume here or CV. You can do that, but go do it yourself and search for yourself. She does not do Ebola research. So this was a cover story, and it's not holding up. This interview she did, so the last time she ever talked to the press in June of 2021, was about 2 or 3 months before DARPA Defuse leaked. So she has not spoken to the press since DARPA Defuse leaked, and her cover story was she was working on Ebola, which made sense at the time. She's in a BSL-4. But here she is in this USGS FOIA being the lead liaison for the animal studies. That basically means experiment with the live bats. So Peter Daszak later came out and said she was playing with live bats in the 1 BSL-4. So that's essentially what Baric was doing. I've done it in other videos. I won't do it here, but he's trying to create a bat vaccine. And that's what Daniel Anderson is going to lead. So one bombshell, Baric has created SARS-2 in 2018. The second bombshell, Daniel Anderson is going to test it on bats in Wuhan. And her story is not going to hold up anymore. So on top of that, her boss resigned as Duke's EID director on January 10, 2020. So this guy resigned the day the SARS-2 genome was published. So it doesn't take a rocket scientist to figure out why he stepped down as the director, because his underling, Daniel Anderson, was inside the Wuhan BSL-4 experimenting with Ralph Baric's genome. So these genomes are basically shared across time and space. Baric's technology is so advanced, it fits in a 1 millimeter tube and can sit on dry ice for 48 hours. And why were they in Wuhan? It was the Chinese horseshoe bats, which is where Baric wanted to try his trial on this novel genome that he had created. And there was more details that came out of this USGS FOIA. And we're still getting our brain around it 2 years later. But inside this detail is something called Baric's ability to create a novel genome like HK3SMEX, which is 20% different from SARS-1, which we now call SARS-2. And he would assemble it with 6 contiguous cDNA pieces linked by unique restriction enzyme sites for full-length genome assembly. Well, guess who does that?
Basic approach that we build coronaviruses using our molecular clone is shown here, with the SARS clone shown in blue. The clone is broken into 6 about 5 kb pieces. Each piece is flanked by bagel 1 restriction endonuclease sites. These are class 2S restriction enzymes that recognize a palindromic sequence, but
notice HKU3 is what he's playing with, and notice that HKU3SMEX is essentially what we see as the final product of what he's created. So Ralph Baric, even going further back into the files here, is basically showing us how we could assemble something like SARS-2 in a 2007 Hong Kong presentation.
Cut and leave asymmetric ends. This allows, since these ends are asymmetric, they actually will not concatamerize like classic sticky ends left by restriction enzymes, but rather they become directional. So if you end clone A with a bagel site that leaves 1 3-nucleotide overhang and the 5' end of the B fragment with the complementary 3-nucleotide overhang, they go together directly. You change different bagel sites at each piece. And this allows them to assemble up into 30 kb chromosomes like Tinker Toys.
Like Tinker Toys. So here's the somewhat famous now preprint that found those 5 restriction sites creating 6 segments. And you can read it on your own here. I'll link to it in the piece. But the Wuhan Institute of Virology does 8 segments, and Ralph Baric always does 6. And guess what? SARS-2 has 6. So that's a big shocker. Now they're here. They're going to order it in 2018 for the DARPA Defuse project, one of the 2 restriction sites that they're going to use to assemble this thing. And going back to Baric, he was asked early in the pandemic in the Red Dawn emails. It was a great question by DHS officials, military guys, biodefense types. These are all U.S. Army, U.S. military bureaucrats. Are there any restriction sites in the strain that are present that are not found in others, especially this thing's engineered? And this is the Red Dawn in March of 2020. And Ralph Baric quickly replied. He hardly ever replies to emails, but he did reply to this one pretty quickly. No, there's absolutely no evidence that this virus is engineered. He didn't mention the 5 restriction sites creating 6 segments, just like SARS-2. Nor did he mention the furin cleavage site, which I'm not going to get into here. So that's just utterly insane. And there was always a question about where Ralph would insert the furin cleavage site, where he proposed to insert the furin cleavage site. And R667 is basically the S1, S2 border of SARS-1. So, and guess who didn't mention the furin cleavage site in front of congressional staff members in February 2020?
Human coronavirus. The genome of the new coronavirus looks just like SARS. Brown is replication genes. Green are structural genes. Gray are genes that regulate virulence. But there's a lot of variation in them, so we don't really know what they do.
Deep sigh there.
This is the receptor shown.
And then he moves on to the next slide. He should have mentioned the SARS-2 furin cleavage site right there on that slide. This is an hour-long technical presentation. He goes on to talk about the receptor binding domain.
And this is the part of this spike gene.
He's talking about details, but he completely whiffed. It was a lie by omission on the furin cleavage site. This is 2 congressional staff in Washington, D.C., in the bowels of the U.S. Capitol building. There may be some senators there for all I know. And this is February of 2020. He is the coronavirus expert that has been called to D.C. from his home base in Chapel Hill, North Carolina, to let us know about this new novel coronavirus that has emerged in Wuhan. And the only thing different between, in the grand scheme of things, SARS-2 and SARS-1 is the furin cleavage site, which enhances pathogenicity, tropism, all these other things, essentially. It makes it, much more contagious, much more lethal than SARS-1. And he didn't even mention it in a very technical presentation. Why? Because he inserted it. It's his baby. It's his genome. It's his pieta. He patented SARS-2 in 2018 and it was leaked from the Wuhan lab in 2019. And here he is in Congress, in the bowels of the U.S. Capitol building, lying about what's inside of it. It's one of the great stories of all time that really no one's talking about. So all I'm trying to do is get the word out my little way possible. So there's a few other bombshells in here I don't really want to get into because it gets a little bit into the weeds. I believe here's another presentation that, here's another presentation that Ralph Baric did with Tony Fauci himself. This is now a month later in March of 2020. And he basically whiffed on presenting the furin cleavage site on this one. And he's basically covering this thing up. So
this results in the following.
He's talking about his book in the 25% deviation, which I might try to avoid in this short little talk. But right here,
coronaviruses.
He should.
The new 2019, the new clade. This is called clade 1. The SARS-related strains appear in clade 3. So these viruses are completely different in terms of any other. We'd never seen SARS coronavirus, group 2B coronaviruses like this before in the past. The closest relative to SARS is 96% identical, which
so what, RA2G13
the last 15 years. The genome organization of the Wuhan coronavirus, or the SARS-2 coronavirus, is shown up here. These are the replicase. The rectangles in green are the structural proteins of the virus, and the boxes in gray are luxury ORFs. And you can see that SARS basically has the identical genome organization. The spike glycoprotein of SARS-2 and SARS both bind the human ACE2 receptor. There's the ACE2 receptor up here in green, and the SARS or the SARS-2 receptor binding domains in the spike glycoprotein down here. And you can see there's a very nice interface between SARS-2 and the human ACE2 receptor. In fact, it makes a better contact with the human ACE2 receptor as compared to the original SARS coronavirus strain. There are 14 contact interface residues in the spike of the receptor.
He just whiffed on the furin cleavage site, but guess who he's presenting with? This is the best part of the whole thing.
Thank you for giving me the opportunity to make this presentation
right
by
behind
video.
Ralph there. It was the subject of my talk in March. We're locked down because of that furin cleavage site that Ralph Baric's inserted into the SARS-2 genome, and that Tony Fauci has funded DARPA Defuse. I don't want to get into that here either. But here they are, Ralph Baric and Tony Fauci giving a side-by-side presentation in this March 2020 during the pandemic. So this is quite the story here. They have created a pandemic virus here, essentially, with what they've been up to. So I'm going to try to wrap it up with the rest of it in here. You can see the links that I'll add to this talk, where you can see where Ralph Baric has created SARS-2 in 2018. Again, SARS-2 is about 20% different than SARS-1 across the entire genome, about 25% at the spike. That's a whole nother talk and a whole nother day. And then Baric basically was asked in testimony, what on earth is that HKU3S mix? And Baric admitted 293 was a typo, which basically confirms what I had written up here already. And I'll link to this piece too. So Baric more or less admitted it, and then he was asked exactly what is it, and he kind of whiffed on it and said, I don't remember. It was 279, which completed his patent that he did. So here's some more updated stuff. Ralph Baric said he would provide this chimera to the Wuhan Institute of Virology for experimental wild-caught bats. This is early in the pandemic. And Ralph Baric even lied, not just to Congress, not just with Fauci, he also lied to the CIA, essentially. He did not declare the furin cleavage site in his CIA presentation early in the pandemic. So he's just lying to anyone and everyone. So here's a piece recently published by Brownstone by myself and Jeffrey Sachs. I'll link to that one. Did Ralph Baric create SARS-2? Yes, he did. Ralph Baric gave 6 hours of testimony to TLDR this one. He gave 6 hours of testimony but couldn't provide an alibi. This is it. This is 6 hours of testimony in 2024, and he could not provide a single alibi. And this was his accomplice over there in Wuhan. Bless her heart, not her fault. She was on the receiving end of a pretty wild cockamamie idea to vaccinate Chinese horseshoe bats with Ralph Baric's novel chimeras, novel genomes, which we now know as SARS-2. So she is more or less patient zero. And I don't want to get into that here, but it's not her fault. We're not to bother her either. But her story just didn't hold up. She was the one in the BSL-4. That mysterious big building had Danielle Anderson in it until November 2019. And there's more on there about cell phones and whatnot that I'll try to avoid in this. But I'm just trying to give you a teaser of what she's doing. She was basically in Wuhan from the June to November of 2019. And she had a cover story of Ebola, and it's not holding up. Because, again, here she is working with Ralph Baric on vaccination of Chinese horseshoe bats. So, Danielle Anderson. And she did not declare this. So she's gone MIA. I don't blame her. I would too. A few other things here. I want to really drive it home that this thing that I call HKU3S mix, which Ralph Baric patented in 2018, that Ralph Baric would provide, using established techniques, we will characterize spike expression and then provide to Lympha Wang. It's, we pronounce it Wang. That's Danielle's boss. And Shi Li, the virus vectors for immune boosting of bats. HKU3S mix. That is the SARS-2 genome being talked about in this 2018 FOIA that was gathered up by U.S. Right to Know. I mean, if you read the U.S. Right to Know, they have a theory that most lab leakers hold to, that somehow Ralph Baric taught Shi Li how to create SARS-2. It's impossible. Ralph Baric said, I didn't. And then they think it was assembled in Wuhan, and it wasn't. And just to really drive home the point about Ralph Baric lying about the genome, I don't have it here, so it's going to take me a minute to pull it up. Why? I want to show you how easy it is to see the furin cleavage site that Ralph Baric proposed to insert. Why publish RATG13? I mean, it was so easy to see that someone like Ralph Baric wouldn't be able to. I mean, there it is. Here's a good view of it right here. So that's SARS-2 at the top and RATG13 at the bottom. It's 100% amino acid match for 500 amino acids around this S1, S2 border, except for that furin cleavage site, PRRAR. And Ralph Baric himself has missed it, a couple of times, essentially, during, many times in his presentation. So I'll link to all this here above. I don't want to go too long on this one because it's a lot to digest. It's better to read than it is to talk about it. But I'm here to tell you with 100% certainty in early 2026, Ralph Baric is the creator of the SARS-CoV-2 genome. But that's only part of the story, because even Tony Fauci and his aerosol specialist in Rocky Mountain Lab got heavily involved. So we'll end it there. Thanks, guys. Take care. Bye.